Okay, next up we have Luca Foschini. How's it going?
Going great. What about you?
Yeah, really good. Thank you. Thank you for coming to speak at SciCon. Really, it's really a pleasure.
In contact.
Likewise. Thanks for inviting me.
Okay, so I'll give a little intro to Luca, and then I'll leave it to you to your presentation. So Luca Foschini, President CEO of Sage BioNetworks. Previously, he co-founded Evidation, where he also led, it was chief data scientist for more than a decade, leading a team of more than 50 health data scientists. He worked with several organizations, NIH, DARPA, BARDA. So I would really say that he influenced the health and data scientist profession. His expertise spans across several fields: machine learning in healthcare, continuous self-monitoring, but is really centered on digital medicine. And he's authored many papers in the field and has worked with so many prestigious institutions like the FDA, NIH, the National Academies of Sciences and Engineering. And I would say, from a personal level, had the pleasure to meet Luca, and I can guarantee that you'll have a really pleasurable experience in hearing his presentation. That said, Luca, I'll leave it to you. Again, really excited to hear what you have to say about open data today.
Yeah, you're setting the expectation pretty high there. So for the next 20-25 minutes, I'd like to tell you a little bit what I've learned, what we have learned at Sage BioNetworks. I'm going to tell you in a second what Sage is. About open data, basically sharing data, mostly biomedical, and how open is not enough, how we come to learn that, and how I've come to learn and internalize it from my dual background. I'm trained as a scientist and as an engineer, and I transitioned to health, as Ricardo was saying, around a decade ago when I started Evidation. So I've been in the shoes of both producers of biomedical data, like Evidation does, mostly patient-generated health data, wearable sensors, and EHR and clinical data. But I'll be talking about genetics today. But I'm also a user of that kind of biomedical data as an AI researcher and engineers. So I'm seeing the struggles from both worlds. And I'm seeing a gap between these two roles of producers and users of data. So without further ado, let's get started. Sage is a nonprofit based in Seattle, around 120 employees that started in 2009. Again, I've been president only for a little more than 8 months. I took office in January. And Sage was started because we feel that science could be doing better, could have a higher yield of conversion between discovery and translation. Whatever comes out of the lab and makes it to the hospital. And this is a great paper from The Lancet in 2014 that kind of dissects the reason why things don't translate as well from discovery to bedside to translation. And the rough rule of thumb that's given in this paper is that of only 15% of discovery of things that are being researched at the very early stage of a pipeline actually make it into something that changes the life of a patient at the very end of the translation spectrum. And that's not because of the intrinsic nature of research, right? Like research by definition, you look at things and you don't know where they're going. So some of those might not turn out into anything. Here we're talking about discoveries. So things that are positively seen as potentially beneficial that just don't get translated because of lack of efficiency, because of lack of clear definition of what is the problem needs to be solved, and having patience sitting at the table when the definition is being created because of analysis that are lacking basic methodological standards, and therefore are even not reproducible. And you can see that each step of this funnel, cuts out a little percentage of potentially translatable discovery that never make it to fully translation. And if that was the state in 2010s, the state in 2020 has not changed drastically in terms of outcome. Still, a lot of discoveries don't get translated, but the why, the how that happens, the flavor of why that happens might have changed slightly. And I would posit now in the 2020s where we are witnessing more of a reproducibility crisis than the other reasons that were outlined in the Lancet paper that I outlined before. This is research from my group. On the left-hand side, we'll look at the reproducibility of machine learning for health research across very basic baselines, like is the code even released? Is the model tested on more than one data set? Are there variance or some sort of error metrics around point estimates? And along those metrics, we pit machine learning for health against more mature fields of machine learning, such as NLP and computer vision. And we saw that invariantly machine learning for health is less reproducible. And that's kind of scary, right? Because when you're thinking about a model that's used in healthcare, you would think that it has a higher risk and therefore it should be more reproducible than other stuff that comes out of research. But machine learning for health is less mature than other more established machine learning disciplines. So it's not too unexpected from that lens, but it's still, quite unsettling that that's the case. On the right-hand side, you see an even more recent work from my group that looks specifically at models to predict COVID from wearable data, which is something that was all the rage a couple of years ago during the pandemic. Everybody was looking at their Fitbit and say, hey, my Fitbit is telling me that I'm sick. So there's a lot of model building that has gone that has happened across many academic institutions over the last few years to both predict COVID or flu based on your wearable data. And unfortunately, a lot of those models are not reproducible. The performance that is cited, is mentioned in the paper, is all over the place. It cannot be explained by dataset differences, at least for the way datasets are described in those papers. And if you actually plot performance against the density of the positives in the datasets, like how many people had COVID in the training set, you see a wide variety, a wide range of different performance at the varying of positive density, which should not be the case because AUC should be independent as a metric from prevalence, but it's not. And so what really this is highlighting is that there are confounders in these data sets that are not being explained. These are really just different definition of COVID that different groups are looking at. And they're just not captured or annotated correctly in the data sets. And so the models that we're developing that we think are detecting COVID are, detecting different kind of severity of COVID that we don't even know how to measure and to compare to each other. So if in 2010, the lack of translation came mostly for the inability to bring the right stakeholders at the table when research needs to be designed and then translated, in 2020, maybe the main culprit is really that the methods that are being developed and the data sets that are being shared are not properly annotated and curated in such a way that different teams can achieve the same results. So that's why SAGE was born, and that's why SAGE continues to be around. It's really to help the scientific community, mostly in the biomedical and health field, to collaborate, share methods, best practice, and really share data as well. We built a platform called Synapse. You can find it at synapse.org. that has supported more than 2 petabytes of biomedical data shared across more than 12,000 research projects over more than a decade. What we have found is that it's really important to coordinate data sharing right at the beginning, at the data producing step, in a very processized way. So we work mostly with academics. We work mostly with researchers that are funded by NIH, the National Institute of Health. And the data they might produce could vary from sequencing of genomics or proteomics or RNA-seq to histopathology or pictures of slices of brains in the case of our Alzheimer work. And this data is collected by multiple groups and needs to be harmonized and needs to be curated in the same way, annotated, have metadata that's consistent across all the groups. And really, the ability to make it openly available to the community is not just a technical problem, it's a social technical problem. You have to build a community around the data sets at the collection time, and you have to think about collection and reuse at the time of collection. So we're creating a playbook that we'll be sharing with the community on how we coordinate this data coordination centers, as we call it, as NIH calls it, in order to make sure that everyone is bringing the same type of data to the table, the data is interoperable with each other, and it's annotated in the same and consistent way. But we've seen that even that is only a necessary but not a sufficient condition for reuse. Yes, you can have a great warehouse where everyone that's a data contributor follows the same schema, and you've curated a community around the data sets such that every different labs that's contributing data to that data set is using the same way and knows about each other. Great. That doesn't still mean effective reuse. That most of the time means that the data is only reused by the subset, the consortium of people that collect it. And so what we've created over time are portals, are a way of making that data more findable, accessible, interoperable, and reusable. FAIR, I'm sure you've heard this acronym many times over SciCon in the last couple of days, through data portals. Data portals, we'll create a different one for different therapeutic areas. And those that really have helped us engage a community around a disease or around a specific, kind of data, mostly associated with a specific disease. I'd love to be able to present you with data that clearly show a causal impact of building a portal in terms of increased reuse. I don't have that yet. It's actually part of my goals in my tenure, being able to measure that in a more precise way. But I can tell you that if we look at one of our longest lived portal, which is the AAD knowledge portal for Alzheimer's disease, and you observe the pattern of data reuse from the very beginning, the first few years to the latest years, you can see that at the very beginning, the people that were using the data were just people around the consortia, as I was saying earlier, people that collected the data were also the one reusing it. But later on, the ratio switched, and more than 2/3 of people reusing the data, downloading the data, were people from outside the consortia. So this is not a controlled experiment. We don't know what the world will look like if we didn't have a portal on it, but I do suspect that the portal really has unlocked ability to bring that data outside of the community that generated it, which I think is a necessary condition for effective reuse. Portals are a good step in the right direction, are a good realization that just open is not enough, but they're not the end of the story. I think they are, again, a wake-up call that make you understand that, yes, you can curate data, make it open, make it shareable, but that doesn't mean that people are going to reuse. You actually have to actively bring it to the community that's interested in the reuse, which most of the time is different, has different knowledge, different norms than the community that generates that data. If you don't do that, then you don't see effective reuse. And physicists in the upper left side of the slides know this already. This is a paper from 2018 of how physicists from CERN have learned that in order to really create effective reuse, you need to not only build data, but also tools for elaborated data and really think about reuse from the get-go. And then on the bottom right side, you have a very recent paper from the MIT group of Lee Antonio Celli that looks at data reuse specifically in the biomedical community and calls out what they call open data in appearance only. Open data a lot of time is called open, but it's really only just openly visible. And in order to access it, you need to talk to someone. And in case of Sage, we're guilty of this too. You have to, most of the time, talk to the governance team of Sage because the data being, health data might have privacy issues, so cannot be, made just downloadable to everyone. The problem is that this governance step, most of the time, is actually put in place almost in a strategic way for people that don't like their data to be reused or don't like them to be reused so swiftly by everybody else and want to have some sort of want to preserve a first mover advantage. I think this is a big plague of quote unquote open data in the biomedical community of like this steps of governance most of the time are much more complex than they need to be. And they really disincentivize people from outside the community, such as, for example, AI researchers to get access to the data. And they make it de facto be open in appearance only. So once again, sharing doesn't mean reuse. We know it very well when we talk about drug development. There's a famous moniker around the valley of death, which is that big gap between academia discoveries of a new target or a new mechanism of action, and then actually industry picking it up and making a drug out of that. And this really comes from the fact that research doesn't work like manufacture. The value chain is not driven by demand all the way back up to the source. Now in manufacture, you have someone that needs a tool, and therefore there's someone that's gonna buy parts from this tool, and people that build the parts will eventually go buy the raw material. And so, the demand propagates upstream all the way to the source. Research is not like that. Research is more like art. There's someone somewhere that solves an interesting problem, attacks a very interesting question, maybe just for the sake of it. And it's just paid for doing that. And they just absolutely only loves doing that. But maybe they do that, from a spirit perspective. They know that eventually it's going to help people, but they don't think about translation all the way. And on the other hand of the spectrum, there are people that are just building translation tool, they're building drugs, and they don't really know what are the big ideas that are being talked about in academia. So you kind of have that gap, and you really kind of have to have someone like, gallery curators you have in art, like people that bring together producers and consumer in a strategic and intentional way that you don't see in many other industry. And so for the last part of the talk, I wanted to talk about one potential channel of building or bringing together two communities that I think should be brought a lot more closer together. And those happen to be the two community which I belong to, the two hats I wear, like the AI in health community and the data producer scientific community. I think a fantastic way of bringing these two communities together, as at least for Sage, has been creating challenges. What are challenges? Challenges are open science events built around a range of biomedical topics where anyone can submit an idea for a challenge, and this challenge should be a benchmark. Should be, it's not a challenge is a data set plus a machine learning task plus a leaderboard. So, for example, find the best way to fold protein, and there is a ground truth that is hitting from the participants, and everybody proposes their algorithm to predict the protein folding of a set of given protein, and the people that do the best win a prize. You can define that throughout for any biomedical research, and they are incredibly engaging. And at Sage, we've been running them for more than a decade and had more than 30,000 participants and written a lot of paper about methods that have beaten state of the art. they are a great way to advance the field on a specific task. And I really think it's the most effective way to generate reuse around a data set is to build a task and create a challenge around that. Just to give some idea of recent challenges that were run on stage, they could really run the gamut between segmenting brain glioblastoma brain tumors. Who does that the most accurately wins, obviously, to guess cardiovascular risk from health record, or understand if a recording of coughing is COVID or not COVID. Again, the sky is the limit. You just really need a well-curated data set and a question that you can turn into a machine learning task, and it can be solved automatically. And there you have a challenge. I'm sure you've heard of Kaggle competitions. What we've run at Sage and the subset of to the Dream community is just like an equivalent of Kaggle, but for biomedical research challenges. These challenges have, in some cases, a clear impact on translation. A great example is the digital mammography challenge, which was done in collaboration with Dream in 2017. Had $1 million cash prize. That's a really good motivator. And it was meant to develop better algorithm to detect breast cancer from mammographies. And we got word that 6 years later or 7 years later in 2023, just recently, 2 of the top 3 winners of the challenge got FDA approval for algorithm for detecting mammography, for detecting cancers for mammography that, we don't know for sure, but we like to think might have been catalyzed, might have been enabled by the challenge that we put together 6 or 7 years ago. Challenges are really also a fantastic educational tool. They are an equalizer. They're able to bring complex scientific question to anyone. And one great way to visualize this is to look at this leaderboard for the CODA challenge, which again is a challenge that is meant to detect whether a recording of coughing is COVID or not COVID, where the third one of the top 3 team is a team of high school students. How amazing is that we can harness the ability, the best skills of anyone out there that's able to put a GPU to a good use and fine-tune transformers in that, regardless of their scientific background. And if you're great at that, but you're still in high school, you can win. You can win cash and you can advance science. So challenges are really API for connecting communities. There's one thing that I think is often overlooked, even if I look at myself, is that community of scientists and community of AI engineers are quite different when it comes to a lot of norms and the way we operate and the way we think about the world and the language that we use. And so having challenges, it's almost like an API. It abstracts all the scientific complexity and therefore enables anyone to focus on their best effort, which is machine learning. I'm going to just talk about one more slide and then conclude. I think that challenges are an incredible way to increase data reuse in the biomedical world, and more generally, benchmark are. A challenge is just a benchmark plus a leaderboard. It's a benchmark plus an engaging component that gets people to engage with the benchmark. But we've all seen in machine learning, in standard machine learning, the power of benchmarks such as ImageNet has been for computer vision that really has started, computer vision in that incredible hike in performance that has brought us to generative AI that we have today. And I really think that we need the same in biology. We need a few ImageNet moments, and I really hope that you can help me make some of those happen. Again, we're running challenges at Sage, but we also are corralling all biomedical challenges out there in this new effort called openchallenges.io. If you know of any biomedical challenge and you don't find it there, please drop me a line and we'll add it to the system. This is a way to centralize knowledge about cool biomedical challenges out there. With this, I conclude, and here you have my references. Drop me an email or Twitter or X or whatever it's called these days. I'll be happy to chat. Thank you again for inviting me.
Thank you, Luca. Thank you so much for sharing a bit about the importance of open data and how actually challenges can help that happen. I have one question about what struck me is that you said open data is not enough, basically. If you don't properly curate, document the data, it is difficult to have what you call effective reuse. So what can we practically do to kind of like increase the chances that others can properly document the data that they post? Is that an education problem in your opinion? Is that an incentive problem? What is that?
Yeah, I think there are two things to talk about. There's definitely the curation and the quality, the perceived quality of data that is important. And as you say, there's no direct incentive for that to happen. Most of the time, maybe here a little bit of context is important. Most of the time, this data is curated by researchers that are paid for their research, not reusing the data. So even if the funder, in this particular case, NIH, has increasingly been strong on pushing for reuse, direct incentive for this curation piece, this like increase of quality that makes it available to other, is not quite there yet. I feel like that the thing to do there, it's kind of just like waiting at this point. The things are starting rolling in the right direction. The NIH is becoming more and more forceful, in a nudging way, in bringing people like us, for example, and bringing Sage as part of a grant in such a way that maybe the researcher themselves doesn't want to curate the data, but Sage can come in. I think there's a huge opportunity there for having a more community-driven incentive. And Ricardo, with ResearchHub, would explore some of this idea of like how can we create incentive for a third party to come annotate data that is not correctly annotated. But I think that is not going to solve the reuse problem by itself. As I was saying earlier, you really have to think strategically about distribution channel. Who are you hoping is going to reuse this data? Is this the AI community? Okay, you have to go talk to them because they don't live in the same space, in the same generation, in the same language than scientists do. They use Discord, right? They don't use emails. They are, they publish in NeurIPS. They don't publish at GA4GH. It's just like you have to actively seek out the community that you want to reuse, almost like in a marketing way, if you will. I think that is the bigger, that's what sets apart, Git from GitHub, right? Git is a tool for sharing code. It doesn't have a social component. GitHub tells you what is the code that's most reused and what's hot in the community right now and what it should be looking at. So we need to create like a GitHub equivalent for biomedical data to the community you want them to reuse to, so people can just like log in and know, oh, the hottest data set now these days is this and that, and I can just download it with a one click from Hugging Face, and I'm done. I think that is the work that needs to be done to really create effective reuse.
Okay, so basically, involving all of this stakeholders, since the beginning, since the creation of the actual, project, the whole idea, starting to talk to people before you actually, create this data, so that what is the best way to produce and share the data.
I think so. I think one good rule of thumb is that thinking that build it and they'll come is not the right mantra here. Right? Build it and they will not come. You can have the best data set out there, and unless you bring it intentionally to the community you want to reuse, it's not going to get out.
Yeah, absolutely. Absolutely agree. I also have a follow-up question that's kind of like more related to what I do. I also work with some type of data, work with biomarkers. And so something that always strikes me when I read, papers is that it's difficult to find reliable biomarkers because there's so many studies that highlight one potential biomarker and then you find other potential candidates. So there's many biomarkers that seem to be great candidates from, for detecting one disease, but very few of these have the statistical significance that you can actually use them to, for example, develop a sensor to detect the biomarker. And then I realized that there are, at the same time, so many studies done in parallel where people analyze, blood, serum, saliva, urines, and so on. And if only there was a way to coordinate and use the same practices on how you collect the samples, analyze the samples, and kind of like increase the statistical significance, that would just like benefit everyone. And so you were talking about challenges. It would be amazing to have a challenge where you bring all of these research groups together to create more statistical power for science. Do you think this is more like, I don't know, maybe that's kind of like utopical, but like, do you think something like this will be, possible in the future where you can all collaborate to actually, make all of this research on biomarkers more effective?
It's so funny you mentioned this. Wasn't even prepared, but I have the secret grand vision of everything could be seen as a prediction task under some lens. So a benchmark, as I was talking about earlier, which is again is a challenge without a leaderboard. A benchmark basically tests out people's algorithm against some sort of ground truth, right? Like your algorithm looks at an image, tries to guess if there's breast cancer there, and then compares behind the scene with the ground truth, is there cancer or not? And if it's right, it gets a score. If it's not, it gets a bad score. Biomarkers are fundamentally measures, and measures are exactly one way of predicting one quantity from another. So you can actually use the same exact process of building a benchmark on biomarkers to bring a community around and develop the best measures that, given some sort of input data, can predict some sort of outcome. I think there are some folks, at least in the digital medicine world, when these biomarkers are coming from digital data, so more like sensors and less so analyte, that are pushing for this. The Digital Medicine Society is really putting out a lot of guidance on how to build measures in a cohesive way, and don't go out and build your own measure by yourself, and because we're all going to do the same thing, and poorly so. Let's join forces. Is one. Human First is a startup that is putting together an atlas of measurements, and really try to embrace this idea of precision measures. But I don't know if there's as much of an effort more on the more bio and analyte world, but I think the same recipe that you would use to have to validate machine learning prediction algorithm, you can use to validate any sort of measures that try to link a quantity to another one. So absolutely, yes, let's build it together.
I mean, absolutely. We'll be talking a bit more about this because I'm just like, as I said, working on this, so I have a few ideas. So we'll be talking more about this for sure in the near future. With that said, Luca, again, it's been a great pleasure. Really enjoyed the conversation. And for anyone in the chat, if you have any any additional question to Luca, just drop in an email. Is more than responsive, and I'll get back to you. Thanks again, Luca.
Thank you for inviting me.